endocrinologist is would they, so what I've done with some of my patients is in consultation with the endocrinologist, we vary the doses. So in other words, it's a kind of standard dose. So I have one patient now who's on like 30% of the recommended dose, and that got her past the complication, the medical complication that she was having with the medication. The other thing that I've done with another child is that we are doing a reduced dose of Zometa, and we just are planning on hospitalization around it. So in other words, instead of doing it as ordinarily would be done as an outpatient, we actually have hospitalization and then we're ready with the appropriate treatment for the medical complications. There's and then there are other medications too. There are other infusion bisphosphonates out there that can be tried, and I'm not the right person to give you that advice. But that's a little bit of kind of generic advice I can give you in my own experience.
Is physical therapy, can we?
Yeah, it's interesting. Yeah, it's like it's right. We're in between a rock and a hard place, right? Because if therapist doesn't want to be responsible for hurting the son on one hand, but then of course we're hurting him if we're not treating him. So I think that and I think that some of the symptoms that you're describing are actually from bone pain, because I think that my patients with and other myopathies, when they get this osteopenia, that they're like my patients with OI, with osteogenesis imperfecta, who have a kind of constant microfracture, and they just have bone pain that is very often is treated successfully with bisphosphonates. So that's kind of like the baseline. I would, this is like mom and apple pie. I mean, I just encourage the therapists to be as aggressive as they can stand, and to keep treating and remembering that without weight bearing, without motion, the bone just gets worse.
Maybe last question. We're running behind.
Probably hoping to ask a question.
Oh yeah.
You spoke about pelvic stability with spinal surgery. It's something I haven't heard of before. Is that something that's quite new, or is that something you've been doing for quite some time? I guess I ask that because Sam has been a wait and see approach for a very long time because of his respiratory status. The concept of growing rods was not applicable for him because there was no way to actually nowhere to fix them, like there was no anchor point. So the idea of the hips is actually something that I've never come across.
Yeah, it's actually pretty standard. We've been doing it for a long time. Technically, I have to say we're a lot better at it now. We were, 10 or 15 years ago. But so even when we're doing, a patient that resembles your son, on our service would be halo traction for 2, 3, 4 weeks, followed by instrumentation, pelvis to probably upper spine instead of ribs. That would be. I'm totally guessing, just looking at it. But it would always be pelvis, and so that leaves the entire lumbar spine untouched. So as we go forward through growth, the lumbar spine and the lower thoracic spine are responsible for more vertical growth than is the upper thoracic spine. So the more we can lead that lumbar spine along, the better we like it, because it gives us a longer trunk height, because we figure most of our patients, their functional height is going to be their sitting height. So we want them to have the longest trunk as possible. But that technique of grabbing the pelvis, even when we're in the growing rod situation, is our kind of, I would argue, is a standard of care.
Thank you.
Thank you so much, Dr. Roy. Hey guys, I think we'll just take a stretch break, okay? Maybe 5 minutes, and then we'll come back with Dr. Michael Lawler once we have his slides all queued up. And we might do 1 or 2, maybe family introductions quick in the meantime. Michelle, Vic, do we have anyone else? Okay.
You want to use your computer?
Sure. I don't think it matters too much, but I know it.
Let me just get this.
So I'll do anything funny on it.
I think everybody, there's about 8 minutes.
Oh, yeah. I don't think
You have a clicker? Yep. Oh, good.
Kid man?
You come prepared.
You guys didn't have a clicker, what would I do? I'd be like, look at all those fibers up there. Some of them are bad.
Yeah.
How's it going? Yes, we talked for a while downstairs. I'll be back when it's done. I hate doing this.
Stage present.
Yeah,
I have it.
whenever you shake it off and start talking, it doesn't feel good right before you do it. Yeah. What the hell am I doing? Yeah. But it never goes when I've been doing this once a week for the past 5 weeks, different topics in front of large crowds, and I still, it just doesn't go away with certain personalities. But we did get through, I think, about all the muscle biopsy reports that we're going to get. I got all the big reports. I got a couple of the other reports that people brought in. So. Yep, took care of his. Did you get it back?
Yeah.
Good. Okay.
You had it.
Yep, I was gonna say
Yeah. And then there'll be a couple we do have a copy of that I can hand. And there may be some people. I'm gonna have people drop stuff off to you tomorrow, or leave stuff for you tomorrow, and just bring back a copy. But I think most of the patients who have muscle biopsy reports available for sharing, either we already have a record of it in CMDIR, or back at home, or I already have it. So it should be all right.
So those people who have it in CMDIR, you guys just telling them to get it later?
Yeah. If they don't have a paper copy. Same thing with the VEX lab, but the VEX lab actually had a copy of all the reports of people that might be involved here, and so I already tracked them down. And
Yeah, not a big one has mine also because they consented to it last time.
Yeah.
So they have
yeah, yours was on the list too, but I had already done yours
Yeah.
with you. So you were late in the list. You had a high number.
I did.
They just moved to Michigan from
Oh, okay. Yeah, I talked to them a little bit earlier. And Kuhn just got here. He's had the stomach flu. He was supposed to fly in last night. 4 of the 5 people in his family had the stomach flu.
Really.
It's pretty good for it.
I'm surprised he came.
Yes. Well, he's supposed to talk tomorrow. You know, but yeah, I think sometimes he lives in Amsterdam and sometimes he lives in Arizona. So I don't know where he's coming from right now.
Maybe Arizona.
Yeah.
It's really hot there.
Yeah, I guess the trip is no treat from either direction, really.
Yeah, we're not in Arizona right now. 110.
I was in Orlando last week for a conference.
I was there in April of this year.
Yeah, I mean, I was walking around the outside the hotel, and I was like, oh, this isn't that bad, this isn't that bad. So I was in the shade of the hotel, and I like turned a corner and like hit direct sunlight, and I almost spontaneously combusted.
Yeah.
That's Florida.
Because it was like, you know, only 97 in the shade or something, and then all of a sudden it was
We're gonna be in Florida, Arizona.
yeah. Well, there were a couple of years ago, a couple of the guys from the University of Florida, they were building up a big muscle center there, and so they're like, oh, what do you think? You know, do you think you might want to come? You know, should we talk to some people about bringing it here?
I know you're right.
No, but now I really think no. Because I don't think I'd last a year there. Yeah.
No, I'm from Canada.
Well, exactly. 30 below zero doesn't bother me, but as soon as it starts topping out 90, you start getting real uncomfortable real
Yeah,
fast.
we were at 90 before that.
Yeah.
Yeah, it was really hot.
Yeah. Yeah, Milwaukee hit that a couple times. Well, you were gone. Milwaukee hit that once or twice last week where it was really hot and humid day.
Yeah, what's in Wisconsin?
Yeah, family.
Family?
Family and cheap land.
I like drove through it apparently. It's like nothing there.
No, Milwaukee's got, Wisconsin has two big cities, and then everything else is.
Are you good?
Yeah, we were in the largest city in Milwaukee, or in Wisconsin. So, like, yeah, so it's like, what did you say, 750,000 people or something?
Hey guys, I think we should probably get going because I want to finish around 5. All right, so I'm stealing Patty's thunder here. She's supposed to be introducing Dr. Waller, but I don't see her. Okay, so we're going to talk about NN's treatment strategies for the next 45 minutes or so. And our next speaker has been a great friend at AFBS. Since completing his postdoctoral research training in the Beck's lab, and moving to the Medical College of Wisconsin in September of 2001, Dr. Waller has continued to work closely with Beck's laboratory while establishing clinical and research neuromuscular pathology labs. The work performed in his research laboratory at NCW has included pathological analyses for a number of preclinical trial studies for animal models of X-linked myotubular myopathy that are currently being performed worldwide, including anti-myostatin therapy, gene therapy, and protein replacement therapy. Dr. Waller is currently working with Audentis Therapeutics to plan muscle biopsy and pathology procedures for human gene therapy clinical trial for XLMTM, and recently has begun collaborating with Solid Biosciences in their gene therapy studies for canine dystrophinopathy. Dr. Waller also has active research projects in his laboratory related to neuromyopathy, congenital contractures, and mitochondrial disease. His work in neuromyopathy is focused on better understanding the disease processes at work in experimental animals, human patients with neuromyopathy. In addition, his lab is currently conducting treatment studies of myostatin inhibition in neuromyopathy mice, which have been supported in part by AFBS, the Muscular Dystrophy Association, and the NIH. Who wrote this? You read this? It's long.
It sounds kind of familiar.
In the spring of 2013, Dr. Laurel's lab became the site of congenital muscle disease tissue repository, which is intended to provide a central place for the donation and distribution of patient tissues. This resource is supported by patient families, industry partners, nonprofit organizations, including Cure CMD, Foundation Building Strength, Joshua Frey's Foundation, and Where There's a Will, There's a Cure. It is our hope that such a central resource for tissue storage and distribution will improve the pace of research in our field. I can also tell you that Michael's one of the most likable guys, and if you have any questions at all, please pin them down, stop them, buy them a drink, all those things. That's great.
Okay, well, thank you very much for that nice introduction. I'm not sure if I wrote that or not. Some of it did sound familiar, but maybe Stacy wrote it. Did you write it?
I didn't write it.
Okay, I don't know who wrote it. It's accurate, except for maybe the nice thing at the end. I will say, if you plan on pinning me down with anything, though, I do have to leave in the morning, unfortunately, and so definitely I'd be happy to talk to anybody after this talk or later tonight. So today what I'm going to talk to you about, the title of the talk is focused on therapy, but given that the time slot of the talk was a little bit longer than I anticipated, I also decided to throw in some stuff about looking at disease processes in amyloid myopathy at the beginning of the talk. So it'll be a two-part talk. I do have a few disclosures. I work in collaboration with Audentes Therapeutics and Solid GT on gene therapy projects, and our research support in our lab is partially obtained from them. I do serve on the scientific advisory boards of Foundation Building Strength and Audentes Therapeutics, and I did until recently consult for Sarepta Therapeutics in the management of their phase 2 and 3 clinical trials for Duchenne muscular dystrophy. So, as I said, the talk today is going to be in two parts. The first half of it's going to be focused on improving our understanding of amyloid myopathy. I'm really only ever interested in treatments, as opposed to a lot of people who are curious about the disease process. The only reason that I study muscle disease is really to pursue treatments. This is a personal preference. I grew up with a kid who is a very close friend who died of Duchenne muscular dystrophy. And if we ever get fortunate enough to obtain effective treatments for any of the treatments or any of the diseases that I study, I will just switch diseases, because it's, as far as I'm concerned, I just want to keep working on effective therapies and improving things on the patient side of things. Now, in the pursuit of that, sometimes we need to know more about the disease. And so I really am trying to push forward several projects that I hope will improve our understanding of disease, be able to improve our treatment trial designs, and potentially use some agents that are, or strategies that are in current clinical use, and improve how your children are treated. So, as you've likely heard about today while I've been downstairs photographing your children inexplicably, this is all focused on congenital muscle disease. Congenital muscle diseases are really genetic disorders of muscle. The two most commonly discussed in forums like this are dystrophy and congenital myopathy. And dystrophies, to make a generalization, tend to be due to mutations in genes that are involved either in myofiber stabilization or myofiber repair. Okay, so patients with dystrophy tear apart their muscles in one way or the other and can't necessarily repair them effectively. And so you have a patient that starts off potentially with relatively intact strength that then damages and can't repair their muscle effectively, and so they lose their muscle over the period of time. Now, in congenital myopathy, you're dealing with a slightly different beast. You are looking at muscle that's not actually built correctly to begin with. So these are mutations in genes that are associated with the process of contraction, either things that are involved in the contractile apparatus that slides past itself to produce muscle contraction, or with the propagation of electrical signals down the muscle membrane that would tell the muscle that it needs to contract. Now, as a result, congenital myopathies result in weakness, sometimes from birth, but certainly usually at a very early age. And it's because the muscle never really works right. However, you're not talking about typically a disease process where you're losing a lot of muscle or myofibers. Okay? And so if an effective treatment can be identified, there is the potential for some significant improvement or reversal of disease pathology. As you can see here, the two that I tend to work in are X-linked myotubular myopathy and amyloid myopathy. A couple of the members of this crowd are on the slide, particularly from a talk I gave in New York City about a year ago. And I always value the opportunity to meet patients and their families. Now, this may be a bit redundant with the prior talks, neuromyopathy is both clinically and genetically heterogeneous. Okay, there's a lot of variability in when neuromyopathy patients present with weakness and how weak they are and what type of care they require. There's also a lot of variability in what's wrong genetically. So right now we know of 10 different genes that are associated with neuromyopathy, with at least hundreds of different mutations, perhaps thousands, okay? So you have a disease that doesn't look uniform, you have genetics that don't look uniform. And so why are these called the same disease? The reason that they're called the same disease is because of the presence of these aggregates of purple junk in the myofibers, or these needle-like rods on electron microscopy, and those are Nemelin rods. And in my clinical side of my life, I'm a pathologist who principally deals with muscle biopsy pathology. And so I'm the type of person that the muscle biopsy goes to. I recognize the Nemelin rods. I say, this is Nemelin myopathy, and then I talk to the geneticist about figuring out what sort of gene it is. Okay, but it's important to recognize that the Nemelin rods may actually not be the problem. All right, and there's a couple of reasons to think about this. First of all, you can find rods in normal muscle with increasing age, but certainly not enough to confuse yourself with Nemelin myopathy. But more importantly, there's really no correlation between Nemelin rod burden and severity. Okay, so you can have a very mild patient with a lot of Nemelin rods in their muscle, and you can have a very severely affected patient with almost no Nemelin rods. And actually, I don't know if Kuhn's going to talk about this tomorrow, but a couple of people in this room have done some very, very interesting studies to show that while Nemelin rods interfere a little bit with contraction, they do not explain the weakness that you see in this disease. And you've got more complicated problems associated with the way the myofilaments are shaped or sized or how they interact with each other. And that really tends to be the issue here. Now, you may have talked about this in one of the earlier talks, but as I mentioned, Nemelin myopathy is a problem, and all congenital myopathies are problems associated with the process of muscle contraction. And so when you think about how you get from your brain to a muscle movement, okay, you start with an upper motor neuron in your motor cortex of your brain that has a big, long axon that goes all the way down to your spinal cord at some level. That interfaces with what they call a peripheral nerve in your ventral horn of your spinal cord. That leaves your spinal cord and it goes to a muscle, okay? It interfaces with the muscle at what's called a neuromuscular junction, and that produces an electrical impulse that is mediated by ions of sodium going from one side of the membrane to the other, and that travels down the membrane, and it goes into these little trenches in the muscle membrane, which are called T-tubules. And that's going to interface with an organelle called the sarcoplasmic reticulum, which has calcium in it. That calcium gets released, that calcium binds to a number of different cellular processes, but also including the contractile apparatus. And the binding of calcium is a critical component in muscle contraction. And then, I think as Alan probably showed you earlier today, you have these thick and thin filaments within the muscle sliding past each other, and that's how the muscle shortens. It's that happening thousands and thousands of times in a given myofiber. Now, interference with any of these things is going to impair your ability to contract muscle effectively. And really, when you're talking about Nemelin myopathy, you're talking about problems with the thin filament here. And so as you can see here, this thin filament structure is a composite of a number of different proteins. A lot of these proteins may look familiar to you because they are all essentially Nemelin myopathy genes. You've got nebulin, troponin, tropomyosin, actin, and cofilin. And when you look at the causes of Nemelin myopathy, the genes associated with it, you are dealing with things that are on this diagram as components of the thin filament, and then other proteins that have recently been found to somehow be associated with thin filament biology or turnover. And so Nemelin myopathy is a disease of thin filament dysfunction. Now, that is a deceptively simple characterization of what goes on in Nemelin myopathy, and it gives you the idea that it should be an easy thing to figure out, and that most certainly is not. When we look at what people have found in different models or cell systems or tissue systems of Nemelin myopathy, there are a number of proven hypotheses that have been put forward that have found muscle dysfunction in nemolomyopathy. And you can see here some of these are associated with certain models and not others, but in some cases it's just because they haven't been tested in all of them. So while you talk about something like the interference with contraction by non-contractile areas or big nemoid rod aggregates, something that could happen in all of them, you can see that you have a real variety of problems that are occurring. You have problems with the length of the thin filament, lack of contractile units. You have some problems of interactions between the thick and the thin filament that need to slide past each other. You have destabilization of critical elements of the thin filament. And then you have the possibility of abnormal muscle repair and metabolic abnormalities. And so this is still a very complex problem, even though we know a lot of the genetic basis of this disease. And this complicates our ability to treat. Now, something to take note of here, there are 4 things that are highlighted in red at the top here. These are phenotypes that actually might give one hope that a non-specific treatment that would just increase the amount of muscle in a person would actually be beneficial. And I'll talk towards the end of my talk about a few trials that we've done in pursuit of determining whether or not this is a useful strategy. So overall, yeah.
What's the example of abnormal sensitivity to calcium?
Well, so abnormal sensitivity of calcium. Kuhn, are you going to talk about calcium sensitizers tomorrow?
Not really.
Terrific.
I skipped it this morning, leaving it for you. What was the question?
Sure. What's an example of abnormal sensitivity to calcium?
So what we have found in our lab is that patients, for instance, with memory mutations, but they need more calcium to generate a similar level of force. And that means that this is not necessarily a bad thing, except that the calcium reservoir, let's say that the calcium bucket that contains calcium in your muscle cell, needs to pump back and forth more calcium to get that more calcium into your muscle cell. And that takes a lot of energy, and that can induce muscle fatigue. So, I don't know if that answers your question.
So what you're saying, now this sounds familiar. I'm kind of Kuhn's wedding photographer on his papers. I don't really understand what he's talking about, but I'm on a lot of them. So the threshold for contraction is higher because you need to go, to contract you need to go from a certain state and then change it dramatically. And so if you're not as sensitive to calcium, you need to either start lower and go higher or just pump a lot more over. And that's going to impair cellular processes because then all of that needs to get put back in. So, okay, so what are the critical questions that we're pursuing? And a component of the critical questions that we are pursuing are determined by our many collaborative interactions with people like Alan, who trained me, and people like Kuhn, who's been a great collaborator over the years. I know what they're good at, and I know what they're doing, and we don't necessarily duplicate efforts. But what worries me is that we have 10 genes, we have a number of clinical phenotypes, and then we have a unifying factor, the nemolin rods, that don't necessarily have anything to do with the weakness. They don't have much. Okay, so are we really dealing with one disease, or are we dealing with a bunch of diseases? Is it adequate to say an ACTA-NM person is different from a Nebula-NM person? Is that accurate? Is that inaccurate? Can we bundle these patients together, or do we need to divide them even further apart? And we are getting close to being able to answer that question, but I think it's a really critical one before we start trying to figure out how to do treatment trials and deciding who goes into what. In our lab, we test mouse models for treatment effects. We need to know how well these mouse models recapitulate the spectrum of human NM, and that is an open question. We'll talk a little bit about some suspicions I have that at least some portion of NM has metabolic dysfunction, and if you have metabolic dysfunction, sometimes you can get better with dietary supplementation. What's the potential for that? And out of the more traditional experimental therapies, small molecules, drugs, things like that, which are effective and which would be the next best to test. Okay, so in our first topic, I just want to talk about determining how many diseases we're dealing with in NM. And how we've decided to focus on it is by looking at protein expression. And although you probably covered this today, really when you talk about expression studies at the moment, you're talking about expression problems or issues at the level of the DNA, at the level of the RNA, or at the level of the protein. And so you have mutations in your DNA that causes problems in the messenger RNA message that gets made, and that ends up causing abnormal proteins, and the protein is the real problematic species here that's acting poorly. Okay, and that's based on issues, mutations in the DNA. Now, it's possible to look at any of these components of cellular biology and at a very large scale and look for abnormalities. At the moment, we are really starting with looking at what we call the proteome. So broad spectrum protein expression throughout the muscle. And the reason we think that's a good place to start is that we have evidence of abnormal protein aggregation in the nemolin rods that we're dealing with here. So we know that we have something that's wrong at the protein level, and chasing that may allow us to detect certain things that may not be as apparent at the DNA or the RNA level. And so in looking at this, we're using the mouse models or the mouse tissue that we have access to. And we have access to mouse tissue from 9 different animal models from around the world. We currently have 3 of the mice themselves in our lab, but frankly, we just need the tissue. And we look for different protein expression signatures. And how we do this, so it's a multi-tiered comparison. Okay, so really we want to look at the animals either early in disease or late in disease. Okay, so for example, if we take one of these models, the ACTA1 H40Y model, which we got from Australia, early in their phase of disease, and we compare them to normal animals, which are called wild-type animals, at the same age. Okay, some things are going to be increased in the ACTA1 H40Y mice, some proteins are going to be expressed highly in the wild-type mice, and some are going to be similar when you compare the 2 mice. Okay, and so in this comparison, you get what I call an early disease signature for this model. All right, you can then take tissue from an animal later in the phase of disease to get a similar late disease signature. And by looking at how protein expression changes in comparison at early and late time points, you can get a signature of disease progression. Okay, and so some of these are going to be pretty non-specific abnormalities that occur when muscle is getting sicker. But the idea is that some of the abnormal protein expression patterns should indicate what's going wrong and what gets worse as the disease gets worse. Now, we can even go one step past this. Okay, we have 9 models at our disposal. We can look at the progression signatures or the early or the late signatures across each of these 9 models, as long as we compare them to the control animals appropriately. And then we can say, okay, we've got 2 models of ACTA1. Do they look at all similar? Do the NEMs look anything like the ACTAs? Do the TPM3s look anything like anybody else? And we can come up with a different means of characterizing these models. It isn't necessarily based on a causative gene, but it is based on what's being abnormally expressed, what's accumulating, what's not being expressed, things like that. We also have the opportunity to identify pathways or processes that are abnormal that we wouldn't necessarily have expected. And so we did this. We did a pilot of this, working very closely with a great collaborator who's now in London named Federica Montanaro, who's a specialist in proteomics. We sent her tissue, or we sent her actually isolated protein from this ACTA1 H40Y mouse and from healthy age-matched animals. And we took this at the late disease stage because we wanted something that, as a proof of concept, was massively abnormal and in order to decide whether or not this was pursuing. And so she found a whole bunch of things. And so she found a bunch of specific abnormalities here. When you look at the muscle-relevant pathways that we kind of got out after really looking at the individual molecules, there's abnormalities of things related to muscle contraction, insulin metabolism, muscle cell stress, protein synthesis, muscle regeneration, actin and actinin protein interactions, mitochondrial function, and acetylcholine receptor localization. And a few of these things, especially the mitochondrial function and the acetylcholine receptor localization, are things that aren't really being evaluated right now as potential causes of weakness in amyloid myopathy, and yet they are potentially treatable issues. Not necessarily curable issues, but at least things that allow some room for improvement using standard nutritional or pharmacological therapy. Now, we also were fortunate enough to contact Eric Olson's group at UT Southwestern, who had done a similar proteomic analysis of the KLHL40 knockout mouse that they published. They gave us the original data. We ran it through the same type of curation that we were doing on our protein. And what we found was we had a bunch of abnormalities that were similar between the two models. We had a set of abnormalities that were unique to the KLHL40s and a set of abnormalities that were unique to the active 1H40Ys. Okay? And so this indicates what you would probably suspect, and that is that when you have these different genes causing this common pathological endpoint, there have to be some similarities. They're all forming amylin rods. And yet, whether or not they're truly causing the same disease really is based on whether or not it's the things in the common pathway versus the things in the unique pathways that are truly the most relevant things to producing weakness here. And ideally, especially since you're dealing with a rare disease, the effective treatment to address the most people are going to be the processes that keep holding up across multiple models and across multiple genes. And those are the things that I would like to target first. So in terms of where we're going with this, and I'll show you an example of where we're going with it in a second. But we need to do protein and gene expression, but mostly protein expression evaluations on the other amyloid myopathy models. This is very time-consuming and very expensive, and I've been beating my head against the wall for about 3 years now to get the NIH to pay for it. They are unexcited about paying for it. There is the opportunity to follow up on our leads using mouse tissue and banked human tissue. Partially, we get this from the tissue repository, and that we already have some pretty good and interesting stuff from that, and I'll show you in a second. And then we're going to identify these disease processes. Some of these potentially have interventions that may help. We can move right on to testing those as soon as we figure out, first of all, what those interventions are, and second of all, which of the disease models that we are dealing with is most likely to respond to it. Because I'd rather not throw the dice and hope that any given intervention works, which is pretty much where I'm at right now with any of our treatment trials. I would rather have information about which model is most likely to respond to a given intervention so that we can give it a thumbs up or a thumbs down in one shot. Okay, so in terms of following up on this, we have all these lists, and these lists get more and more intimidating as you get more of it. But as I said, you want to isolate things that are common between the different models. And something that really stuck out to me is mitochondrial dysfunction. And part of this is because I can understand how mitochondrial dysfunction would be understudied. Studying mitochondrial disease is awful. I happen to study mitochondrial disease because I had a bunch of geneticists drag me into it at MCW, and I have to diagnose it all the time in the clinical lab. So, we have some projects that are based on improving diagnostic testing in mitochondrial disease. But really, when something like a mitochondrial disease signal comes up, it's a very variable phenotype. It's very difficult to really interrogate the mitochondria. And so this is something that may be a target of opportunity that might have been left by the wayside. And mitochondria, as I'm sure many of you know, are the real powerhouse of the cell. This is an EM picture of mitochondria. They actually look a lot like they look in a high school or a grade school textbook. They look like round or little kidney bean shaped things. And then they have a pretty characteristic folding pattern of cristae in the center of them. And when mitochondria are bad, they are shaped poorly, they are too big, they are too convoluted, or they've got inclusions in the middle of them. And so diagnosing mitochondrial disease when it's severe enough to produce morphological abnormalities actually isn't that bad. But what complicates things is that sometimes the energy dysfunction does not actually produce morphological abnormalities. The other thing to keep in mind is that mitochondrial abnormalities can be primary or secondary. Okay, you can have mutations in genes that cause the mitochondrial energy production to be poor. And so this is going to be your cause of disease. And yet, you can have diseases that interfere with mitochondrial energy production as a secondary effect. And that's not really the primary problem. However, I would say that if that's in secondary in a disease, as I suspect it is in some forms of NM, it's still a potentially treatable issue, and it's something that we should be looking at. So, to further contextualize why I'm a little suspicious of mitochondria, I just want to show you what mitochondrial disease looks like in an adult biopsy versus what enamel myopathy biopsy from a patient that's in this crowd looks like. What you may see is that they're not necessarily hard to distinguish from each other, especially when you take an adult versus a kid. And really, I don't have a problem diagnosing one versus the other, but what you should notice is that when you look at the real characteristic stain people keep showing you on amylin rods, which is the Gamorre trichrome stain, the mitochondrial aggregate looks like a big red clump of garbage in your cell, and an amylin rod aggregate looks like a big red/purple clump of garbage in your cell. Okay, and so when these are pure disease states, when you've got mitochondrial proliferations versus amylin rods, they look different. Okay, but what happens if you've got both? Would you notice a mitochondrial problem? And I don't think that a lot of pathologists would. I don't think that I did necessarily until I really started looking. Now, a way to distinguish this is by looking by electron microscopy, looking to see what the mitochondria look like. And as I showed you, this is what a normal mitochondrion looks like. And these are all examples of mitochondrial disease. This one's got big, crazy-looking mitochondria. This one has a proliferation of mitochondria that actually don't have enough complexity. And then these three all have different sorts of inclusions within mitochondria, all of which are going to interfere with energy production. When you look at some of the amylin myopathy models, including this H40Y model that we talked about, and a human autopsy with ACTA1-related NM that I did personally last year, you've got amylin rods, they're the yellow arrows, clearly amylin myopathy. But you also have a bunch of really funny-looking mitochondria. There's too many of them there, they're shaped wrong, and while these look blown out and this is post-mortem artifact, even if you put Humpty Dumpty back together again and made them the normal size, there's still too many of them there and they still look bad. Okay, so there's some evidence for at least secondary mitochondrial pathology here, and it is an area that is also supported by our proteomic studies. So as I mentioned earlier, looking at mitochondria is difficult in terms of diagnosing a primary disease. Okay, we tend to do it due to the lack of gold standard diagnostic tests through a three-strike approach, combining clinical history, light microscopy, electron microscopy, mitochondrial electron transport chain activity, and mitochondrial DNA content. However, in this case, we're not really looking for abnormalities of, say, the amount of DNA in the mitochondria. We're not trying to diagnose mitochondrial disease. We're just looking for abnormal function. And so what we started doing is sending out specimens for electron transport chain activity. And this is just a diagram from a recent book article that we did that shows what's called the electron transport chain. It shows how different complexes within the mitochondria move protons. Those protons then are sent through a pump, and that's how you generate ATP. ATP is how you get energy in your cell. So abnormalities within the mitochondrial electron transport chain can cause problems with this and problems with ATP production. And there's a specific clinical assay done at CITEM that can evaluate at least the first 4 of these. And so what we're doing right now is we've just sent out 3 sets of tissue on the 3 animal models we have, the 2 ACTA1 models that are in our lab, and the nebula conditional knockout. We sent them to CITEM with the appropriate controls, and we've asked them to do the clinical grade mitochondrial electron transport chain testing. And this bill for this is entirely supported by AFBS in a generous pilot grant support. Now, if we have any indication of mitochondrial dysfunction, frankly, in any of these models, we're going to go to the NIH with this. And we're going to present an R01 grant based on biomarker identification with the proteomics and the gene expression, and then follow up by chasing down mitochondrial disease. And the component of this is going to be looking at nutritional supplementation of the animals in a way that you would use for clinical mitochondrial disease patients. And frankly, another element will be to look at the impact of poorly understood treatments that seem to be effective in some NM patients, such as tyrosine. You don't really understand what it does, but I wonder what it does to energy metabolism. So things that we don't understand that we're currently using, this may actually provide some degree of explanation for it. Okay, so part two, I'm going to talk about how we are starting to evaluate treatments in a disease that I've just admitted we don't know nearly enough about to do this. Something that you're going to hear more about tomorrow from Alan is how the genetic complexity of nebulomyopathy makes the design of genetically based treatments problematic. And so just on the topic of this, if you look at your two main causes of nebulomyopathy, two main genes involved, you've got nebulin and you've got actin-1. Okay, you've got two very different problems here. And when we talk about gene therapy and some of the other congenital myopathies, like X-linked myotubular myopathy, they're very different problems. Okay, X-linked myotubular myopathy produces disease by essentially not having any myotubulin. Okay, nebulin myopathy isn't really necessarily produced by deficiency state as much as it's produced by an abnormal activity of the proteins that are abnormal in it. Now, when you look at the types of genes and proteins you're dealing with, even in comparing these two, there's a lot of variation. Okay, nebulin is enormous. Okay, 249 kilobases. Different muscles treat the gene differently. Okay, and there aren't any real mutation hotspots. So there are strategies for dealing with large genes that are mostly based in the dystrophin gene. But the dystrophin gene is a little bit easier on people. So it's not quite this large, and it definitely has spots that tend to get mutations versus spots that don't. Okay, so that means you can identify certain regions that if you were to manipulate the genes there, or the expression there, that you would be targeting a significant proportion of patients. And frankly, that doesn't happen for nebulin. You've got an enormous gene, and the mutations are spread all over the place. The other thing to note is that many patients with nebulin-related MM have two different mutations on their two chromosomes, and so you have to take genetic manipulation strategies very cautiously, because there's a potential for when you fix one of the chromosomes to actually induce a mutation in the other one. Okay, so these are problems that aren't really present in the genetic manipulation studies you guys may be seeing or talked about in the congenital myopathy community, and they're things that we're definitely struggling with, trying to figure out a way to plan them. Now, in terms of the actin-1 gene, it's actually small enough to cram into an adeno-associated virus for gene therapy. But the real problem is that, once again, it's not a complete deficiency state. So actin is part of the spin filament, and you can see that they're just kind of arranged as beads here. Okay, and actin interacts with other actin molecules, actin interacts with other proteins. All right, if you have abnormal actin, it may prevent the binding of the actin to itself. It may prevent the binding of actin to other proteins. But adding more normal actin may not be incredibly beneficial if you've got a bunch of bad actin ruining the party for everybody. Okay, and so at this stage of genetic manipulation therapy, people are still trying to establish safety in something that people consider highly risky. You know, this is a reason that there's a little bit decreased enthusiasm for starting out with something like these, where you may accidentally make people either underwhelmingly better or worse. Okay, so there are a variety of potential therapeutic approaches in nebulomyopathy. We're starting to kind of untie them. As I mentioned, gene therapy and protein replacement therapy tend to be more useful for genes and proteins in complete deficiency states, at least at this stage. It doesn't mean that it won't be useful after we get a level of comfort with those technologies. Things like gene silencing, people talk about for dominant negative effects. Alan's going to talk a little bit about CRISPR tomorrow, so I'll leave that to him. There are strategies being used for Duchenne muscular dystrophy, like exon skipping or stop codon read-through. They might be useful for a subpopulation of mutations that cause premature stops of frame shift mutations, but that's a relatively small amount of patients. And really, and as you can tell by my copping out from the calcium sensitization issue earlier, while calcium sensitizers and troponin activation is a potential therapeutic option, I'm leaving that to him, and I'm happy to take pictures for him. So what I'm left with is myostatin inhibition and contracture management. That's what we're going to talk about. So, okay, what's myostatin? So myostatin is something that prevents your muscles from growing too big, right? Because while having a ton of muscle is a great thing for beach season, it's not really energetically efficient. Okay, so animals in the wild don't need enormous muscles. And look how ridiculous that cow looks. It's a ridiculous looking cow. Okay, it's not even as functional as a regular cow. All right, it's muscle bound. It's enormous. Okay, but it's not necessarily as functional. Same thing goes with that racing dog. And both of these are myostatin knockouts. So having a knockout of myostatin allows your muscles to grow to essentially their level of physical limitation. And the pharmaceutical industry has been pursuing agents that allow this to happen as a treatment. Okay, and we've been dealing since about 2008 with Accelerant Pharma, which has been a wonderful collaborator, and now we've branched out to a few other collaborations in the field. And for most of what we've dealt with, we've used a drug called R2B MFC, because that really rolls off the tongue. That acts as a receptor decoy for the myostatin receptor, so it soaks up whatever myostatin is in there and any other ligands of this receptor, prevents myostatin signaling, allows big growth of muscles. So we started out doing this in X-linked myotubular myopathy mice. It did work to grow the muscle. It didn't work to improve strength, and I think that's because excitation-contraction coupling is bad in myotubular myopathy. That's why we went the protein replacement gene therapy route. But we did think, based on a lot of the work done by people that are in this room and outside of it, that myopathy may be a better fit because it doesn't have the same types of excitation-contraction coupling problems. And we thought, okay, we can take this drug that makes normal animals really big and really strong, apply it to a different disease state without the limitations we've identified, and hopefully match things up. And so we have done 2 trials with myostatin inhibition in myopathy, and we started a 3rd one last week, each using a different model. So the first one we did was using this ACTA1H41 mouse that I've been showing you information on. And these animals have a point mutation in ACTA1. They're generated in Australia. We chose them for our first trial because half of the male animals died from disease in the initial characterization, and it is appealing as a person that wants clear treatment outcomes for an animal model severe enough that they die from their disease. So you can look and see whether or not survival is improved or death is delayed, and there's not a lot of equivocation about what that means. We did use this ACTR2B MMC in hopes of improving muscle growth and strength. And so we did this trial. It was published a couple of months ago, and it was significantly supported by AFBS and a few other funding elements. What we found is when we treated normal wild-type animals that haven't been treated, or they've been given the vehicle, are in blue here. The treated ones are in orange. Okay, the wild-type animals got bigger and they got stronger when on this 4-limb grip strength testing, which is kind of it's kind of like a measured pull-up for a mouse. Now, when we look at the ACTA1H40Y animals, the vehicle treated are in green, the purple are the treated, and they did get heavier, but they didn't really get significantly stronger on the 4-limb grip strength. So we were kind of underwhelmed by that, kind of disappointed. There's no real difference in how well they ran around, but the interesting thing was looking at the survival. Once again, this was done on all male mice because we wanted to capture the survival phenotype. We had the previously described problem with survival in the male mice, in the vehicle-treated mice, and yet we didn't encounter death nearly as often in the treated mice. So we had an improvement in survival, not only to their lifespan, but even to the amount of animals that died. So that sounds great, right? My problem is that as a part of my clinical expertise, I know what things died of, and these things died of something funny. Okay, so we looked at all the organs, we looked at all the muscles. The muscles didn't look particularly bad in the dying animals versus the non-dying animals. And frankly, functionally, as we were testing them, they didn't look that bad either. What did look awful was their genitourinary tracts. Okay, so essentially, they died because they had urethral outlet obstruction. It turned into a UTI, and then while their bladders didn't explode, it got awfully close. They died from complications of that. Okay, so you can see here with a normal prostate, urethra, and then there's some skeletal or some striated muscle around it, a normal animal. You can see here, there's the muscle, there's a big dilated urethra, big dilated bladder, all kinds of necrosis and inflammation. Looks awful. Okay, so unfortunately, this forced me to learn a great deal about the mouse genitourinary tract and mouse reproductive behavior, which I can never unlearn. But it did come in handy. Okay, so when you look at the way a mouse plumbing is set up, you've got urethra, you've got prostate, and you've got a whole bunch of striated muscle around it. There's actually a lot more striated muscle than you get in people. The whole thing is different. You also get this big concretion in there, which mercifully is not a feature of human biology. And this is a component of the reproductive behavior. So when a mouse mates, it shoots this rock into the female, and that's evidently improved something. Well, when you look at the animals we had in this study, okay, just using skeletal muscle actin staining on the striated muscle that's surrounding the urethra, you can see that wild-type muscle, as you would expect, has pretty even distribution of actin. And yeah, when you look at the H40Y muscle, you've got these big clumps of actin, you've got these big actin aggregates. The striated muscle surrounding the urethra has some very, very significant myopathy pathology, likely impairing its function. I also can see a lot of inflammation. And when we quantified these things, we didn't really find much of a difference in copulatory plugs when you compared at least some of the wild types to the H40Y animals. We did find a massive difference in inflammation when you think about it. So when the animals die, when you're taking them down, they can shoot out these copulatory plugs as kind of an agonal, their dying response. But the inflammation is a good track to what was causing problems before they died. It's a tissue reaction to something being stuck there, okay? And when you look at how many of these animals had inflammation, none of the wild-type animals did, treated or untreated. Almost all of the vehicle-treated H40Y animals had some evidence of genitourinary inflammation consistent with bladder outlet obstruction or urethral outlet obstruction, and this was massively decreased with treatment. And so while this treatment didn't produce better pull-ups in these animals, it did seem to produce some element of improved striated muscle function so that they were not obstructing their genitourinary tract, probably with these copulatory plugs. So where were we at the end of this thing? So obviously I was totally grossed out. I'm a huge germaphobe and this was just too much. But our treatment improved survival, and it was based on striated muscle function, but it wasn't necessarily based on the prevention of the types of phenotypes that we see in patients. So the usefulness of this model, the usefulness of this phenotype is questionable. We did find this weird GU phenotype that we need to keep an eye out for as we look at other things, and overall the benefit was uncertain. And so we moved on to looking at a different mouse model using a different drug. The reason for the different mouse model is because we have no real confidence that even when you compare the two different ACTA1 mutant models, they are really caused by the same thing. They are different point mutations in the same gene, okay, but these 2 point mutations do something very different between each other. And also we changed the drug mostly because the NIH told us that they wanted us to use a more exciting drug than the thing we've been using for a couple of years. We formed a collaboration with Pfizer, and we started working with this drug. And this is actually the first time anybody's talked about this. I got clearance to talk about this at 8 a.m. this morning. So this is hot off the presses for you guys. So what happens when we did this? Okay. There are similar findings here where with animal weight, so animal weight gets better in both healthy and the ACTA1 D286G animals. But now we actually have an improvement to grip strength. Okay, so you see 2 overlapping lines here, you see 2 overlapping lines here. But what's actually happening is that your untreated wild type and untreated ACTA1 D286G animals are pretty close in terms of their ability to do a pull-up at baseline. Okay, these are weak animals, but they're not necessarily weak with respect to their ability to do a pull-up. When you give them the myostatin inhibitor, they both get better, and they both get better by about the same amount. Okay, and so that's very, very interesting, because you would expect building more bad muscle to really produce a submaximal response. And yet what we're finding is it's just as good at increasing strength in the wild type animals as it is in the D286G animals. Now, when we look at elements of strength where these animals are deficient, there are only a few very small things that we could check. We did a ton of different tests on them, and in most cases, they're not particularly severe. It didn't change rotor rod performance, which is kind of a rod's rolling around. And they got a, it's kind of like one of those lumberjack things where they're trying to stay on the log. If you don't know what I'm talking about, just nod, it'll make me feel better. And then this is like a voluntary running wheel thing, like a gerbil wheel thing. And it didn't actually affect either of those. But really, this is a different component of strength that you are talking about when you're talking about bulking something up and giving it better burst strength. And so the grip strength was something we were really hoping for more than anything else. The interesting thing here, though, is that we have two different active on models of disease. We have two very different responses to the same therapeutic type of approach. I don't think this is a difference in the two drugs. Frankly, if anything, the Pfizer drug is more selective than the Acceleron drug. The Acceleron drug, that class had unacceptable side effect profiles in early clinical trial testing, which is why they made something more specific. But there's no reason it should work better. It's just safer. And yet, you have much better improvement of forelimb grip strength and consistent forelimb grip strength in this model in comparison to that model. Okay, so that tells us that even the gene that's associated with is not necessarily predictive of treatment efficacy. So what are we doing? So we're evaluating the pathology, and then we're also looking at the muscles from each of these two studies to see if we can figure out if there's any indicators of what might confer treatment sensitivity versus treatment resistance with this sort of strategy. All right, I mentioned a third study. This study started exactly 1 week ago, last Saturday. This is using Dr. Henk Renzier's nebulin conditional knockout mice, using another new myostatin inhibitor. And I am very hopeful that this will also be beneficial. These mice look like they're very good for therapeutic interventions. A fair amount of them die in a certain period of time that is useful for our treatment studies. They die at about 2 or 3 months of age. And the animals have severe pathology. They have small fibers. There's a lot for us to fix there. Based on the last 3 or 4 drug studies that we've done, I anticipate we'll be done this in 8 months. And so by the time I talk to you next, I imagine I'll have something to tell you one way or the other. So a comment that I've kind of, or an area of comment that I've kind of insinuated a few times here is, a critical question here is whether or not we're even choosing the right models for our study. Okay, if we had just looked at the Ecto-1 H40Y model and then thrown the whole idea of myostatin inhibition in the garbage, we would have said that it was useless. I don't think that it's useless. I think it's useful for a subpopulation of patients. I think we need to know more about the disease and what these models represent before we can identify those patients. But certainly, we've got more evidence of functional benefit for nebulin myopathy mice than I've seen in a lot of the other studies here. And so we need to know more about what we're dealing with. Now, there's also the issue when we're doing these models of how valid they are. Okay, so there's issues what they call construct validity versus face validity. So construct validity refers to a model that is generated by the same means as the disease. So an Ecto-1 H40Y mouse has high construct validity because that mutation style is based on seeing human patients with Ecto-1 H40Y mutations. Okay, they're caused by the same thing. They may not look anything alike, and that's what face validity is. Okay, whether or not the disease looks like itself. So these Ecto-1 models have very good construct validity, but may not look an exceptional amount like the disease. They have a few extra pathological changes that you don't tend to see in nebulin myopathy patients. Whereas things like the nebulin's conditional knockout mouse is a conditional knockout that starts knocking down nebulin at a certain point in time. Okay, so that looks a lot like nebulin NM. It has good face validity, and yet it's made on a molecular basis by a completely different mechanism. Okay, and so that makes it really suitable for certain non-specific strategies like this, and pretty unsuitable for genetic manipulation strategies because your genetic problem isn't quite the same as what your patients have. And so, as we look more at things like the protein expression patterns, disease processes involved, generalize this to how much of these issues are really present in the human tissue you guys are donating to the various repositories. These are all questions we can answer. These are all questions that I think we can answer soon. Okay, as one final thing, because I know that contractures affect the nemolizumab population, I thought I would talk about our work in modeling congenital contractures in mice. And contractures actually occur in a number of disorders. They're not necessarily all neuromuscular disorders. So you can get them in certainly severe muscle disease, severe nerve disease. You can get them in cerebral palsy, so brain injuries or strokes that occur in utero. You can get them with oligohydramnios, so when there's not enough amniotic fluid or when the amniotic sac is not large enough. The critical factor in producing congenital contractures is actually the incapability to move while you're in the third trimester of fetal development. Okay, you can be motion restricted or you can be motion incapable, but the muscle tendon unit needs to mature in the third trimester, and it needs to grow a certain amount of muscle, a certain amount of tendon to be totally functional. And if you don't, you get short muscles, you get long tendons, and that's what actually forms a muscle contraction. And these can be lifelong sources of disability, and currently the only treatments that people really use are variations of stretch therapy and surgical intervention. Now, the curious thing, although the term is thrown around excessively, in doing a simply awful number of human autopsies in my period of practice, and a lot of mouse autopsies, these don't actually happen in mice. So people talk about deformities and contractures in mice. They don't really have this muscle shortening, tendon lengthening. The mice just kind of look crappy. They call it contractures. And the reason for this, I think, is because the muscle tendon unit matures at a different rate in mice in comparison to humans. When you take a human that's day of life at term, okay, that's not the same muscle tendon unit as a mouse at the day of life, at the first day of life. It's more like a mouse at 3 weeks of life. Okay, so there's a postnatal maturation of muscle tendon unit in the mouse that does not occur in the human. And so I got together with Danny Riley, who's a great collaborator at MCW, who evidently didn't have enough to do, so we got to go for this. And said, well, what if we just restrict motion on a mouse when it's young? Can we produce contractures? So that's what we did. So we used the cast in our proof of concept experiment to immobilize the ankle, starting at 1 week of life, and we took it off at 3 weeks of life. And then it had a stable deformity that we tracked it for 2 months, and it didn't go away. We looked at what the muscles and tendons look like. Okay, this is the unsplinted limb, and this is the immobilized limb. You can see tendon lengthening, muscle shortening. And when we did some measurements, we saw that you have this tendon lengthening and muscle shortening in the muscle tendon units that only cross the immobilized limb, and the things that cross both the knee and the ankle didn't get them. So the activity that was present at the knee was enough to prevent the contracture. So we said, okay, well, we got this, and actually it was fairly reproducible. I was shocked. This thing I expected to fail 8 ways from Sunday, but I wasn't really doing it, so I didn't care that much. So we said, okay, let's improve the cast design. Okay, because we don't want to necessarily start out at full plantar flexion. That gives us only one direction of motion, and we want to be able to test the impact and stretch in multiple different muscle groups. So we came up with a couple of different cast designs. We settled down as a 90-degree cast, which would allow us to manipulate things, either moving the foot up or moving the foot down. So plantar flexion or dorsiflexion of the paw. And in the ability to do that, it gives us the ability to stretch out either the soleus muscle, which is on one side, or the tibialis anterior muscle, which is on the other side. And there are reasons in terms of how these muscles are built with respect to how many fast and slow fibers they have, that it's nice to be able to look at these things. We did some tests of these casting strategies. They all seem to work out pretty well. So what we ended up doing is using this 90-degree, and then when we wanted to look at stretch therapy in our pilot experiments, we took animals that had contractures, and then we serially casted them to make them more plantar flexed every few days to stretch this TA muscle. And so this is what we found with the 90-degree casting alone. These are scale drawings by a wonderful artist that works as a technician in my lab, who also evidently wasn't working hard enough one day, so I made her do this. So we look at soleus muscle, impact of inducing the contracture with the split. You have a displacement of the soleus muscle, shortening of the proximal tendon, lengthening of the distal tendon. It looks a lot like the contracture muscle. Tibialis anterior muscle doesn't really have a proximal tendon. Then you have this tendon lengthening muscle shortening that I showed you in the prior setup. So at that point, we thought we were in a good place to start looking at treatment. We used this plantar flexion serial casting to look at the impact of stretch. And so this is the same information here. And by inducing steady stretch by 2 weeks worth of casting and progressively more stretched out lengths, we actually had less muscle shortening and less tendon lengthening. So we had a partial therapeutic effect to stretch therapy. It's highly reproducible and pretty well reproduced what people know from human studies. So now at this point, we actually have a good model in which therapies for contraction can be tested. We had an idea in terms of looking at electrostimulation to improve the response that totally did not improve it at all. It was essentially identical to what the stretch alone looked like when we combined it with stretch. But now we have a system in which we can start looking at pharmacological or PT type interventions on contractures, see if we can improve the efficacy of stretch therapy and potentially improve function. Now, things that we need to evaluate, so we are in the process of evaluating what drug that has some promising results. We need to look more specifically at what this looks like microscopically. We need to make sure it matters on a functional perspective. So look at how strong the muscle tendon unit is, see if making longer muscle, shorter tendons is actually conferring additional benefits functionally. And then look at why this is occurring and see if we can make some good educated guesses on what therapeutic options will be good things to test next. So overall, we're pursuing a variety of projects related to NM. I'd like to think that all of them will directly contribute to our future treatment trials as we continue to answer our questions. I'm really interested in looking at mitochondrial dysfunction and actively looking at it with the help of AFBS, and hopefully soon the NIH. I think that that's a low-hanging fruit that if we can identify something even in a subpopulation of patients, we might be able to functionally improve people with some pretty minimal interventions. And then we're really focusing on myostatin inhibition trials right now. And that has some promise, but there's a lot more work to be done, and really we're working as fast as we can. So I work with a lot of people. This is a very long list of a very big amount of places. For a lot of them, I just try to do pathology. We have a lot of generous supporters, some of which are represented here, and a wonderful lab in both our clinical and research labs. And thank you very much. Sure.
In your research, are you eventually going to get to a point where there's a pill or an injection to fix that thin filament that's defective? Is that what you're striving for?
I would like to think that eventually we'll get to that point, because that's going to be the kind of maximum level fix to fix the primary problem. I think that given the complexity of not knowing, there's so many problems wrong there, that at the moment I am chasing after the low-hanging fruit, like making the muscles bigger in hopes of making them stronger, to make people more functional as we learn more about what we're dealing with. But yes, that's the eventual goal.
Is that in the near future, or are you talking many years yet?
I think the non-specific benefits will hopefully be in the near future. So I think the things that will hopefully improve function by 10%, 20%, maybe keep people off ventilators, maybe, make communication easier and things like that. I'd like to think those are in the near future. I think that for more permanent fixes, you're probably looking at more genetic manipulation technology. And for a disease like NM with its complexities, I see that as a second round of diseases as targets of opportunity. After these things are established as safe, diseases like myotubular myopathy, and keep in mind the plan at the moment is for myotubular myopathy to hit clinical trials in the next year or two. Once we get a safety record with that, this is certainly a disease that I'm talking with all of the 10 different drug companies I work with, keeping it on the radar, talking about ways that we can use the drugs that they're doing, therapies that they're doing, to repurpose it for NM. But I would say that a considerable limitation at the moment is the fact that while we have animal models, we don't know enough about how well they relate to human disease.
If you get good results from the mouse trial in 8 months, what are the next steps?
Next steps, so these are drugs that are being pursued for Duchenne muscular dystrophy and other causes of muscle loss. So the next step is to say, okay, well, we've got great data here. Discuss with these drug companies the potential to pursue a human clinical trial in the NM population, or if they are at a stage of the human trial process where safety has been established for these agents.
They wouldn't go right from mice to humans, right? You would go to like dogs or something like that?
We could go right from mice to humans. Technology. But without intermediates. And so it's an issue of being able to monitor closely and then have it washed out. So even when we were doing mitochondrial protein replacement therapy, the regulatory environment for getting it approved would have been much different because we weren't sticking a gene in there that was going to be there for a long, long time. We were sticking a protein in there that would degrade and get peed out or whatever. So you watch them closely, but you wouldn't. It's not an irrevocable decision. So yes, there are drugs that go from mice to humans, and this is something I would support if this continues to be useful. The other thing is that while there are endomyopathy dogs that get reported, there isn't a colony available for testing. So you wouldn't necessarily stop drug testing development based on the lack of an animal's existence for testing.
Like, how good are the development of human muscle tissue but in the petri dish, right? Like,
Yeah.
I'm talking about using urine to actually develop.
Yep.
Yeah. And then, has there been any other areas of study where they're using those sort of fabricated human muscle cells, tissue to actually come up with treatment?
So that would be more like a cell therapy based thing. And this was actually, this isn't a specialization of mine. I was on one of those urine papers, but once again, I was more the wedding photographer than anything else. The use of engineered muscle tissue was more of a big deal when people were afraid of using viruses to deliver genes. So the idea behind that was that you would use immature muscle cells, myoblasts, that have nuclei that have the intact genes. You would inject them, they would fuse with the muscles, and then you would get some expression. Okay. The problem is that you have to do a lot of injections and generate a lot of cells in order to treat a person, and then they won't go very far. Now I think, and that was more viable as an idea when people were terrified of using viruses to do delivery. Now I think we're at a point where viral delivery is a real thing, and that I don't really anticipate the types of catastrophes that occurred in the late 90s associated with gene therapy. So I think the usefulness of that in a disease that affects every muscle in the body is limited.
Okay, thank you, Dr. Waller.
All right, thank you very much.
We are going to do the Unity Walk tonight. I know everyone's tired, so if you aren't up to it, we understand, but we are going to go out and just do a quick walk around.
Currently, we do it twice a week.
Grab some people in the kids' room. Again, no pressure if you want